Welcome to the Laboratory of Genomic Instability and Diseases. Our team investigates the mechanism of DNA replication in higher eukaryotes, both body-east and human, and we try to implicate its role in various diseases. So our major focus areas in DNA replication, there are two aspects of it. One is the basic research and another one is the applied research. In the basic research concept, we try to understand the mechanism of DNA replication in human and yeast. There are several DNA polymerases they express in the cell and they try to compete with the other DNA polymerases and they try to occupy the DNA strands. So we want to understand what are the cellular phenotypes of these DNA replication mechanisms. Our approach has been to understand that if a pathogen replicate in the host, it will cause diseases. So we are trying to identify such attenuated strains for antifungal vaccine development, as well as we try to target some of these components for antifungal drug development. We engineered a FOL32 mutant called CNA25. In preclinical models, this mutant was both non-pathogenic and immunogenic. It induced robust protection against multiple Candida species by activating innate, adaptive, as well as trained immunity with TLR2, Dactin1, TH17, interferon gamma, and TN-alpha and involvement. We demonstrated that EDTA-treated Candida albicans is also attenuated and it also protects from the reinfection. As a NACI Fellow and AMI Award recipient, I am deeply invested in training the next generation. We are leveraging CRISPR technology and genomics to find out novel replication factors. We are engineering attenuated fungal strains for broader vaccine development. Our lab embraces innovation, rigorous experimentation, and social impact. We welcome researchers and collaborators passionate about DNA biology and fungal pathology. Let's come together, push the boundaries for genomic medicine.
I study the role of DNA polymerase kappa or TLS DNA polymerase in chemoresistance of OSCC. Polymerase zeta is known for its involvement in the cisplatin resistance. However, my study revealed role of polymerase kappa in the cisplatin resistance of OSCC by rewiring DNA replication, which is Fall-Kappa variant.
My work further strengthens the implication of cos32 knockout NUCA as whole cell vaccine candidate, as upon deletion of Fall32 given in hypervirulent strain leads to apprehency and gives protection upon reinfection.
As we know that the stability and the functionality of tRNA depends on the modifications present on it, so our lab has launched a project that aims to understand the role of different tRNA modifying enzymes on tRNA stability, protein homeostasis, as well as fungal pathogenesis, and currently I am working on that.
My study focuses on studying the molecular mechanism of protective immunity generated by a vaccine strain, CNA-25, and improvising the routes of vaccinations.